Also learned about abiotic versus, or anaerobic versus aerobic. So, yeast are aerobic organisms. Anyone know what aerobic is? There. With oxygen. So, they like air. They like to multiply in the air. But, if you notice in these, you seal them in. So you can actually induce them to grow in anaerobic states. If yeast are growing aerobically, they're not producing alcohol. They're going through their entire glycolysis process. But if you seal them off, all of a sudden they go, making alcohol is my best shot at getting any energy. So these are, you can, facilitated anaerobes. So aerobics with oxygen, anaerobe is
Without.
without. We also learned about, pasteurization for Pasteur. I mean, that's why it has that name. The entire idea of heating something to kill all the microbes and then only putting in what you want. Questions? So, moving to a different branch of microorganisms, diseases. So, if you can't see them, how do you know that they're causing people to get sick in hospitals? I mean, this was one of those big things. They didn't even know about microorganisms, so, some people died. It was one of those things where it used to be, you wouldn't know how to blood type somebody. They've got red stuff, they've got red stuff. Why is it sometimes it helps the patient and sometimes it kills them? Just some of those things you didn't necessarily know. So, microorganisms can cause disease. So we have Knock, who demonstrated the relationship between a microorganism and disease. He postulated that there's a series of steps to show specifically that this microorganism causes a disease. And it ended up having a lot of applications here. He was going through, he was studying, he was isolating, but it kind of led us to the Semmelweis and hand washing to reduce the spread of infections. So now this is really a sad story. He was noticing that in this teaching hospital that the moms that were getting their kids delivered by his teaching students died a lot more often than the ones midwives were delivering. And he's like, what's going on? So his idea was, you know what, they're down playing with cadavers, whatever cadaver dust is on them, they're then taking into the birthing rooms, and that's what's killing the moms. So we can look at it now and go, that's kind of silly, but then again, in a way, it's right, They were carrying bacteria with them. It ended up being they were actually carrying staph, which was on their hands, which we all have on us, which is fine, except for if you get in an open wound. So he started insisting on hand washing. So he had his students hand wash. And all of a sudden he got, I think the death rates were dropped from like almost 20% down to like 1 or 2%. But because it's such a weird thing, cadaver dust, what are you talking about? He got laughed at. And even though he had the numbers, they just kind of sent him packing, going, no, get out of here. You're just ridiculous. And you're like, saving people. Obviously something's right here. So, but because he did have those numbers, later on, they were able to actually look at this and go, he was on to something. Let's wash hands. Sounds good. So we got Lister, who developed some antiseptic techniques. Because a lot of times you cut into somebody, they then get gangrene or they get some infections. And so, it's like, if we kind of clean our surgical equipments before we use them, maybe sterilize our treatment bandages, we get patients who live a lot longer. And was able to reduce a lot of the post-surgical infections. It's like, okay. Also with he had such a major impact on basic microbiology because he was the one who really started studying anthrax, which is a rod-shaped bacterium. And so he was actually able to study and look at colonies of these microorganisms. From him, we have simple staining techniques. The first spectromicrographs, so the first visualization of some of these microbes. Techniques for figuring out how many colony-forming units, CFUs per mil. So figuring out doubling times and how much is in a vial. Being able to use steam to sterilize. Also, Petri dishes came out of his lab, as is most things. So one of the people working with him was Richard Petri. And so anyone know what a Petri dish is? If you don't, you will by the end of this class. So just those the little dishes that we use all the time, have media on the bottom, have a lid, making tons of Petri plates. But just that concept of having that little plate with that lid came out of his lab. Back in 1887, also figuring out ways to sterilely transfer bacteria from one place to another. So one of the things you guys will be doing in this lab, or in this class, is doing we have an open air lab where you set something out, and you see how many things you can get to grow. Well, if you wanted to study one of those colonies, how can you, sterilely select just that one colony, transfer it, and do all those techniques came from him. Then I realized this was in there twice, so we won't. Or I was going to say. So, from all of this basic microbiology research, started all of these other fields. Eller had this great, he wanted to find a magic bullet. He's like, can I find something that's going to treat everything? This is kind of where chemotherapy came up. I mean, chemotherapy, the general idea is it's chemicals used to kill. So trying to selectively do things. He came up with an arsenic derivative that was used to treat syphilis. We got immunology from all of this. So Jenner is very famous for his smallpox vaccine. When you look into the history of science, it's not always very ethical. Something we're working with more now. Jenner is a great example of that because he was noticing, hey, all these people have smallpox. These milkmaids don't have smallpox, but they have all these weird blisters on their hands. So the milkmaids were getting cowpox from the cows, and the cowpox was making them immune to smallpox. So he decided to take a volunteer who was an 8-year-old child and intentionally give him cowpox. And then go expose him to smallpox and say, hey, look, you didn't get it. Great, it worked. So, not necessarily the most ethical, great way to do this, but in general, immunology came out of this. It's also why vaccines back a cow due to the cowpox. Another big field that came out of microbiology is nursing. So, a lot of times you hear about Florence Nightingale, nurse. I never quite really understood where that came from. But what it is Nightingale was one of the nurses, she was a head nurse, and she started going, you know what, this is ridiculous. These hospitals are filthy. This is not the way this is run. And she changed everything. She started getting everything washed regularly, off-site, get things sterilized, have cleanliness in the hospital, antiseptic techniques, make it so that all of a sudden these hospitals became clean places with fresh bandages and sterilization. Yeah.
Didn't she pick up those ideas training in Germany?
Probably. I don't think she came up with it, but she was the one who forced it to be happening.
I believe she trained in Germany and picked up the ideas there.
Probably, because she's not the first one. I mean, the hand washing came up from other people before her, but she's really known because she was the head nurse, and so she made all of her underling nurses do this. And so, if you all of a sudden have, it's a regime change. This is now how we're doing it. So, not a lot of patients in her hospitals live longer. Epidemiology, so study of occurrence, distribution, spread of disease in populations. Trying to figure out these cholera experiments. I mean, come on, London, the reason why they left all the time is because they would just throw all the sewage out, so everyone would get sick every summer. So, trying to figure out the study of all of this. These are all just different branches of studying microbes, or working with them. Then also, we need to know how are we going to defend against disease? How are we going to defend against these microbes? So we have serology, the study of the blood serum. You know, blood contains chemicals and cells that fight infection. Immunology. So how does our body respond? Not just how can we initiate a response, like with vaccination, but how do we specifically target and fight different pathogens? Chemotherapy, so penicillin. This is where we kind of discover that idea of like Escherichia bacteria, you get this other kind of bacteria growing, and all of a sudden your E. coli dies. You're like, why did that die? This is where you start getting some of our different drugs came from microorganisms. Let's see. What else? Basic chemical reactions. Biochemistry, the entire field of biochemistry, where we are in a test tube studying what's going on. That all came out of this. So, where you can do, there's in vivo and in vitro. So, anyone know what the differences are? In vivo, in vitro? I'm sure you've heard these.
Inside the tube.
Like in vitro fertilization?
Outside, then you put it in.
Outside. So, in vivo, in life, it's inside of a cell. In vitro, in a test tube. So being able to study things in a test tube, start to break them apart and look at them, which is where we get, we can design herbicides, pesticides, different, we can start diagnosing illnesses, getting different tests for drugs, and monitoring diseases. We can also start figuring out, what about the genetics? You know, now that we know yeast produce alcohol, why? How? What genes do this? And then, so what are the microbial genetics? What are the molecular biology of all of this? And then, the more we know about that, you can start doing your recombinant DNA technology. So how many's heard of GMO? No one's heard of a GMO. I was going to say, come on. It was a proposition recently. Genetically modified organisms. So, taking something and changing it in the lab. How do you do recombinant DNA technology? How, you know, there's a lot of people whose lives have been benefited by this because it used to be insulin, or was it human growth hormone? Human growth hormone had to be isolated from the pituitary glands of cadavers. So, hard to get. And it's coming from a cadaver, which means you could pick up a human virus. Now, by being able to take that gene, put it into E. coli, let E. coli produce it, all of a sudden you can get mass quantities, really cheap, and it's clean. No viruses, no cadavers. And so, recombinant DNA technology gets a bad rap, but a lot of people, you know, insulin, we get through recombinant DNA technology. A lot of our drugs, we get through recombinant DNA technology. Gene therapy, something else we learned from all of these different studies of microbes and everything. How can we try and change these genes? How can we try and do gene therapy to make things better? Okay? Chapter 1. Good. Any questions? I move fast, so stop me when you have questions here.